Fragile X syndrome is a genetic disorder that can cause autism spectrum disorders. People with Fragile X have large, protruding ears, an elongated face, large testes in males, and significant behavioral problems such as repetitive movements. They also have intellectual complications, including social anxiety and developmental delay. Fragile X arises from the disruption of the FMR1 gene on the X chromosome. FMR1 is the Fragile X mental retardation 1 gene that produces Fragile X mental retardation protein, or FMRP for short. FMRP is required for normal brain development. In the FMR1 gene, an excessive repeat of the CGG triplet in the 5' untranslated region causes Fragile X. In normal circumstances, we have 5 to 44 CGG triplet repeats. However, in Fragile X, the affected individual has over 200 repeats. In response to this unusually high occurrence, the gene is methylated and therefore silenced. Since males only carry one copy of the X chromosome, the symptoms associated with Fragile X are more prevalent. In normal individuals, the CGG repeat region is often interspersed with AGG repeats. To understand the mechanism of Fragile X, we want to know, does the absence of AGG repeats within the CGG repeat sequence confer instability? We will conduct this experiment using mouse as the model organism. Step 1. Design a vector targeting the repeat region of the endogenous FMR1 gene. Step 2. Insert the target vector into embryonic stem cells using electroporation. Step 3. Select for cells that are GAN-cyclovir resistant. This means that the vector has successfully integrated into the genome via homologous recombination. Step 4. Inject cells into a new mouse embryo with a different coat color. Step 5. The resulting chimeric mouse is then crossed with a wild-type mouse, and among the offspring are mice capable of passing the CGG repeats to their own offspring. Then we observe the phenotype of the mice over successive generations and monitor repeat expansion. If we see expansion of the repeat region and Fragile X symptoms, then we can conclude that AGG repeats within the CGG repeat region may confer instability.